Okay, let's start. Shall we start? Sorry for the slight delay. I hope you had a good break. Let's start. So we're going to continue on the cytoskeleton. Yeah, if you intend to stay, please take your seat. On the cytoskeleton, jumping into microtubules right now. Okay, so before the break, previously I told you about how the cytoskeleton of cells, including actin, microtubules, and so on, is really essential to determine the shape of cells and basically their polarity, their structural polarity, and therefore their functional polarity. And we talked briefly, at least, about the enterocytes. We're going to talk more about migratory cells. I already talked quite a bit about actin. I hope you did not forget everything. I'm going to try to remind you of some of the things, the key concepts I mentioned before the break. And today we're going to really focus on these other two kinds of elements, mostly microtubules, and a tiny bit about intermediate filaments. There's a lot of ground to cover today. So briefly, because we've seen this before, we're still in introductory. So I told you about actin microfilaments, and there are a lot of similarities with the microtubules I'm going to highlight. The idea, one first idea, that although this is a fibrous higher order complex itself, and it's true for the microtubules, the monomers are globular. And the fibrous structure emerges from polymerization. And that process of polymerization is what confers dynamics, what's heavily regulated, as we see. So both polymerization, actually, as well as depolymerization. So that's true for actin. We've seen quite a few examples. It's also true for microtubules. Although the monomers, they actually come as dimers, and these are dimers that are being added to form the tubules, and I'm going to talk quite a bit about that today. A key difference with intermediate filaments, besides the fact that there are various kinds of proteins, is that the monomers in intermediate filaments are themselves fibrillar proteins. They are not globular. Okay, so it's almost as if inherently they are more about structure than dynamics. Sorry. So a brief reminder about the lecture on actin, mostly to give you an anchor to compare with microtubules. We've seen it binds ATP, and we talked about thread myelin, which is one way to harness the energy of ATP and convert it into mechanical force. The polymerized versions come in many different forms. We talked about networks, about bundles, and we've seen some key differences in how actin dynamics is going to be regulated to produce these structures. And we've seen, so some of the key differences we're going to see is that actin, we've seen that previously. There are many places in the cell where actin is being polymerized and depolymerized. So the dynamics of actin is very local. And we're going to see that's a difference with microtubules here. And in all cases, it's the filaments are highly dynamic, they polymerize, depolymerize. They are polarized, that's essential. We've seen that. There's a plus end where polymerization predominates, and a negative end where it's rather depolymerization. They tend to serve as tracks for dedicated motors, the myosins, and you've seen the myosins actually walk on actin filaments using atomic force microscopy. And yeah, they are the main part of the what's at the cortex of cells. So if we use this as a reference point to look at microtubules and try to identify both shared properties as well as key differences, here's how it goes. So microtubules, they are found throughout the cell, but as you may already appreciate here, there are places devoid of microtubules at times, and there are places where it's highly concentrated. And whereas for actin, we've seen a lot of the concentration at the cortex and in the leading edge of migratory cells. For microtubules, they tend to concentrate slightly more around the nucleus. And I'm just going to use, it's not 100%, true, but I'm going to use this as an anchor to get to a key difference in how they are regulated. But in both cases, they are highly dynamic and polarized, and we're going to talk about that quite a bit. In other words, each microtubule has a plus end and a negative end, just like actin filaments, and both microtubules and microtubules like actin, they tend to be polymerized and depolymerized, and that regulating that dynamics is essentially what the cell does to determine the distribution of microtubules throughout the cell. Now, they can be, unlike actin filaments, they can be really, really long. So a key example, a classic example is the axon of a motor neurons of a whale has to connect the spinal cord all the way down to the tip of the tail. So it can be you can have really, really long microtubules there. And one simple way to remember kind of a difference is that you may see microtubules as sort of highways, going long distances and maybe not so many of them, whereas actin filaments would be more your local roads and local tracks, if you will. Okay. So other differences is that they are made of tubulin, as we'll see. They are a bit more rigid and don't bend as easily. And a very important point, their assembly is regulated for a small number of locations, which are called microtubule organizing centers, or MTOC, and we're going to talk about MTOCs over and over again. And then, in terms of motors, there are tracks for different kinds of motors, two, actually, kinesins and dyneins. And one of the key points I'll need you to remember about these is that kinesins tend to walk towards the plus end, and dyneins walk towards the negative end. And that's going to be essential to understand their roles in cells. And again, whereas actin is involved in the contractile machinery, and the cortex microtubules, they tend to be involved in long-range trafficking of vesicles and organelles, right? So I know many of you were dying to know how these vesicles move about. Well, it involves microtubules quite a bit. There's one thing special, and I'm not going to talk a whole lot about that in this course, that microtubule, they tend to be the underlying cytoskeletal architecture for very specialized structures or even specialized cells. So here are a few examples you may be familiar with. Ciliae, for example. So you guys know where are essential ciliae? Actually, no. In the intestine, the microvilli are actin-based. But that's good because I wanted to highlight that difference. So microvilli, very tiny, actin-based, actin bundles. Ciliae tend to be longer. There are some very important ciliae that allow you to listen to me right now in your ears, right? So those are the kinds of ciliae. Actually, this picture is from the inner ear. Very important roles for microtubules in this amazing structure, the mitotic spindle, right? So, and this is a place where I'm not going to talk about that much in this lecture, but when cells divide, almost all of the microtubule in the cell are now rearranged to make the spindle, right? So imagine if you're a cell that migrates, you organize your microtubules in a certain way, or an epithelial cell, in a way to traffic organelles and so on. But then at the moment of cell division, the entire microtubule cytoskeleton is reorganized to make the mitotic spindle. Other unique and important structures involve the axon, of course, of neurons, and we're going to go back to those at the end to illustrate trafficking of vesicles, trafficking of organelles in long distances in the axons. And the flagellum of spermatozoids, for example, or other organisms allow them to swim around and so on. So these microtubules, they can be used to build a great diversity of structures at this kind of higher order level. But then when you look closer, they all have a very similar type of organization. And that's going to be a more essential focus of today's lecture. So this is an electron micrograph, and you can see the scale here. And if you were to look at this and extract some information, what would you say? What do you see? Is there order in this?
Yes.
Yeah, there's some kind of order. You have, so if that's the matrix, the medium, there's kind of a lumen, and then it seems to form a ring, and you have these bigger units that are repeated with some kind of radial symmetry. Right? Okay, so fairly simple. So this is what's sometimes been referred to as a singlet, but to understand this, it's actually best understood as a cross-section through a microtubule. Okay? So what this is showing is that you have to make the tube, so you have the inside is kind of empty, and you have 13 of these protofilaments here. So protofilaments is now, if you look in the other dimension, a string of those beads here. Okay? So each one of those elements here would be one bead in a cross section. Okay, so that's an essential organization of the microtubule. So you have 13 of those protofilaments. Actually, a more useful way to memorize the basic structure is to think about them as 13 protofilaments organized in a way that they make a tube. Okay? And then I'm going to highlight right away an essential point here, which is going to be this seam here, right? So before I tell you about the subunits, if you look at this diagram, it's very well done, because you notice that the protofilaments are slightly offset in an x direction. Okay? So it's not like if you were to try to model a microtubule with a sheet of paper, you would not fold it perfectly horizontally. You would create a slight angle. See what I mean? So that creates a seam here where the interactions between the two protofilaments across the seam is a little suboptimal. And the reason why I highlight this is because this is providing a structural instability, right? It's kind of an imperfect assembly. And we're going to see that this is extremely important for the dynamics of the microtubule, this sort of intrinsic instability of the microtubule. But besides that, there are, of course, like I was saying, polymers. So they are built from monomers, actually dimers of alpha and beta tubulin, repeated here. So you see the dimers are the ones the dimers are the ones to be free floating, okay, and they are the ones to be added or recovered when the microtubule depolymerizes. Okay, that's the building block of the microtubules. It comes as a dimer already, so those are two different proteins encoded by different genes, and as you can see, they can bind GTP. And this can lead to hydrolysis of GTP. So when you have, again, a structural element that can bind GTP, polymerize, depolymerize, at some point you might imagine there might be a connection between GTP hydrolysis and harnessing the energy of it to create force. So from this, you can also see, if you look at one protofilament, you see each dimer is added in this direction, with the beta tubulin sticking out on this end and the alpha tubulin sticking out on the other end. So by analogy with actin, this is also providing a polarity, right? And so classically, the plus end is the end where the beta tubulin is the last subunit, and the negative end is where the alpha tubulin is. Okay? When you take a close look at this molecule, so indeed there are two globular proteins. They both can bind GTP or GDP. You see that here the GDP binding pocket or GTP binding pocket of beta tubulin is close to where the next alpha tubulin would come. And just as an aside, it was determined that the drug Taxol, which has been a drug used to fight cancer quite a bit, derived from a tree, binds here, and basically what it does, it stabilizes microtubules. So I'm using this again to give you a taste of what's coming, which is you have something that has to do with microtubule dynamics, and that's related to the beta tubulin and its structure, and actually, as we'll see, its ability to hydrolyze GTP. Okay? I'm going to get back to that. So that's the basic building blocks of microtubules, these alpha beta tubulin dimers, and how they can be added. So unlike actin, like I said already, microtubules are built from only a few places in the cell. And so here I'm showing you a couple examples. So this is a cell in interphase, and you see there's a high density of microtubules here, and they seem to radiate towards the cortex of the cell. The nucleus would be here. In this kind of staining, that's an old staining a postdoc in my lab did. So this is a place where you have a lot of a higher intensity of microtubule binding protein, and it also tends to irradiate throughout the cell. And in dividing cells in the spindle, again, you have things coming out from here and then reaching out, right? So this is very different from actin. It's a key difference I want you to sort of really grasp. Actin, you can nucleate actin and create filaments pretty much anywhere in the cell. It depends. What does that depend on? What's the key kinds of protein that will regulate that? I know it's Monday morning after the spring break. No. Well, yeah, effectors of what? Rho GTPases, right? The most important slide of the whole course. Come on. Okay. So MTOCs, what's up with MTOCs, right? So there, I'm kind of building some drama here to make you think about and pay attention to this essential difference between how microtubules are regulated, how their dynamics is controlled, compared to actin. So in cartoon forms, what I just showed you, you have the centrosome here, which is a key microtubule organizing center. I'm going to touch upon the centrosome a little bit. And then you have microtubules sticking out from the centrosome and reaching out all the way to the cortex. That's a cell in interphase, like a migratory cell. A key point is that the negative ends are not even showed, but they are around here. The plus ends are sticking out. Okay? So, and as we'll see, the plus end is really the business end of the microtubules, as we'll see. And so it's a very similar logic in spindles, where you have now two centrosomes. Actually, centrosomes need to be replicated before mitosis. So you have now two centrosomes, which each organize half of the spindle, and then you have microtubules coming and somehow talking to each other and interacting with chromosomes at the kinetochores to position them in this metaphase plate. And then here again, the negative ends are near the centrosomes, the plus ends are sticking out. Okay, and I can show you yet another example and another example. Almost wherever you look, you tend to have localized microtubules organizing center and plus ends sticking outwards, right? In a centrifugal kind of way. There are some exceptions in dendrites of neurons, and some special structures like the basal body in cilia or flagella have different kinds of organization. So here the microtubules are, have these so-called doublets or triplets here. I'm not going to bother you with this, just some specialization of the microtubules. We're going to really focus on what's going on in these cells. But before that, so everything starts in this microtubule organizing center. Okay, so let's take a closer look at this. So they are really, really peculiar in some ways, and still being studied quite a bit. So just going to give you a very brief overview. But the peculiarity of the centrosomes is that you have these two centrioles here, which are relatively short, and you always have two positioned at about 90 degrees relative to each other. And they are surrounded in this electron-dense, so-called pericentriolar material. When you look, if you have proteins that, you use a protein that accumulates in this area, it's going to stay in here. So that's really where most microtubules come from. You take a closer look at a cross-section of these centrioles. They are made of tubulin as well. And you have these triplets, and now you have usually 9 triplets organized in a very specific way, and they replicate each other, and quite frankly, quite a bit of their biology is still to some extent under study. But the key point I want to highlight is that they're embedded in this pericentriolar material. So in some ways, you could see this as a membraneless special place area in the cell where you have a high concentration of pericentriolar material, including this gamma Turk. So that's what I want you to pay attention to. So gamma Turk contains gamma tubulin, and Turk stands for gamma tubulin ring complex. When you stain for gamma tubulin in the area of a centrosome near a nascent microtubule, as shown here, you see the gamma tubulin is on the one hand here. And like I was showing you, the negative ends are all sort of anchored or rooted in these MTOCs. Right? And the previous cartoon shows how they are in this pericentriolar material. So the whole idea, simply put, is that gamma Turk gets organized, so it's a tubulin as well, so it gets organized in this sort of one full turn of a bit of an helix, and that's the template to start building a new microtubule. Okay, so if you will, it's analogous to the ARP2/3 complex when we talked about actin, which served as a template onto which new monomers are added. Okay, so here that's exactly the idea. You use gamma Turk as the template in the pericentriolar material where there's a high concentration, so polymerization is favored, and then you can start making a microtubule. Okay? So studies, I'm going to go quickly through these 2 slides because actually they are about almost the biophysics of microtubules, but they do not reflect very well what happens in cell, and I'm going to insist on this. So you could do very similar studies to what was done with actin, trying to determine the critical concentration of dimers needed to add to this gamma Turk, to add to existing filaments and promote polymerization. And you would see that indeed, again, by analogy with actin, the negative end where the alpha tubulin complex is versus the plus end, there's a greater propensity to add dimers at the plus end. Okay? But somehow, the dynamics at the negative end in cells is irrelevant for microtubules because negative ends are held in gamma Turk. Okay, so the only place where microtubules are regulated is at the plus end. Okay, so that really begs the question, which is how do you modulate synthesis versus or polymerization versus depolymerization? Okay. So that's why I'm going kind of quickly through this, because in principle there is a room for treadmilling also with microtubules, but in practice, and that's the main point here, the cellular concentration of tubulin is so high, so much higher than the critical concentration, that everything is regulated by proteins. Almost nothing is due to the biophysical properties of microtubules. Okay? So let's look into that. How are microtubules made, right? So far what I've told you something about the structure of microtubules, their polarity, their composition, the structure of the tubule with this imperfect seam, and this idea that they all come from these MTOCs, and it starts for individual microtubules with the gamma Turk ring, right? So now the idea is presumably dimers are going to be added to the gamma Turk, and that's how microtubules are going to grow. Okay, so let's try to look at the dynamics. So here is a simple experiment. You kind of old school in some ways. You take cells which you keep cool, so nothing really happens. They have a pre-existing microtubule cytoskeleton to begin with. And then you add fluorescent tubulin, And so this loads, nothing happens until you switch the cells back to 37 degrees, and then you see addition of tubulin on pre-existing microtubules. Right, so unlike actin, if you did that with actin, you would see de novo filaments. Here, the newly synthesized microtubules is predominantly on pre-existing microtubules. Okay, and in a movie form, so this is how it looks. Damn. So look at these guys. What are they doing? Yeah, they are growing. Are they just growing? Growing and shortening. Right? This is the key. If you want to, okay, let me be clear. You have to walk away from this lecture with more than one thing. But if there was one thing, only one thing you walk away with, that's this dynamics. The dynamics at the plus ends of microtubules involves polymerization, growth of the microtubules, and from the same microtubules, depolymerization. So let's look into that. So indeed, in fixed, you would see, for example, here A is growing, and then eventually, from a few minutes later, shrunk. B just keeps growing, and C doesn't do much. Okay, so if you plot these dynamics for individual microtubules, this is how it looks. It starts where the microtubule is growing. So here you see time and the microtubule length as a function of time. And of course, this is a simplified diagram. So we've seen polymerization. So length increases. But then eventually, almost invariably, especially when you have dynamic structures like a lamellipodium and so on, you have deassembly. And the tipping point has been referred to as a catastrophe, right? And we're going to see why. We're going to try to explain what happens during these catastrophes, when microtubules just depolymerize very quickly. But then eventually, there's a rescue. So the depolymerization stops. Now you have a plus end where you can add again some dimers, right? So that's the rescue, and now the assembly can continue. And really, the life of microtubules can be described in this way: a succession of catastrophes. I mean, it's kind of poetic in some ways, right? A succession of growth, and until a catastrophe hits, and you depolymerize, and then you get some rescue, and so on. But that's just a microtubule. Don't get too emotional. And it's a normal part of their life. And that's an intrinsic property that's being used by cells to regulate them, to regulate the whole cytoskeleton as a whole. So when you take a close look at these microtubules under either elongations or polymerization or catastrophes, here is how it looks. So this is now a side view electron microscopy, and because it's slightly more dense, you can somehow recognize individual protofilaments, right? So if you look back at the cartoon, this is a side view from a tubule, and then you have the protofilaments. And then under a catastrophe, now it looks different. So you see, it's very blunt end, okay? Now under a catastrophe, it's very different. It looks like, and you could remember a palm tree, a banana you're peeling, or whatever, but it really does look like you're peeling individual protofilaments. Okay? So the key here, if you just look at this image and try to think about what's going on, is that you see, you can still recognize individual protofilaments, but now they are bent. And because of the bending, they peel off away from each other, right? So so this is part of the depolymerization. The deassembly of protofilaments. So in other words, you deassemble protofilaments before you even deassemble dimers. Okay, so this is different from the actin case, where dimers are being chopped away. And this is another reason why I was insisting on the seam, right? Because the seam was an imperfection in connecting protofilaments throughout the microtubule. So the seam structurally promotes this behavior, this peeling of protofilaments. So in cartoon forms, this looks like this. When the microtubule is growing and dimers are being added at the plus end, it's blunt, it's straight. Then And okay, now this is the key switch that every year people are like, okay, so how does it work really? Well, the key question almost every microfilament is asking itself is, if you think about the plus end, you have now 13 beta tubulin, and it turns out, I didn't say that yet, but the dimers have GTP bound beta tubulin. Okay, so if the 13 protofilaments are still have GTP bound beta tubulin, the structure stays put and new protofilaments can come. But if before the addition of the next dimer on any of the 13 protofilaments, GTP hydrolysis, so the beta tubulin hydrolyzes or whatever, there's GTP hydrolysis of beta tubulin, of the last one. Now it causes a change of conformation. And now you can start having that one protofilament peeling off. And when one protofilament starts peeling off, it triggers a chain reaction almost. That's the catastrophe. The other filaments start peeling off and bending in this way. Okay? So, and this is why you have a quick depolymerization of the negative ends. So then you might wonder, so what happens when you have a rescue? Well, you need to somehow chop or remove those bent protofilaments and recycle these dimers, and this is what happens here. And then you can have the reassembly. So of course, all of this is extremely regulated. I'm just showing you what happens at the level of microtubules. Okay, so this is kind of an important slide, right? Because it's the molecular mechanism behind this dynamic instability that is assembly, polymerization, until, and it's kind of a stochastic event. Now you have a catastrophe and rescue, etc. But it provides dynamics to the system. Okay, because indeed there's very little growth from random places in the cell. So in this experiment, you start with a cold cell, and now instead of adding, before you add the tubulin, you treat with colchicine. So colchicine is derived from a plant, and what colchicine does, it depolymerizes microtubules, right? But you still have the MTOC, you still have the centrosome here. So now if you start adding, and that you can see here with some microtubules still sticking out. But now if you add fluorescent tubulin and switch back to 37 degree, again, this is the only place where microtubules come from. Okay? So in these kinds of experiment, what will determine, okay, bear with me, because where I'm going is, okay, by combining this question of having one place where microtubules are growing from with this dynamic instability, now we have almost everything we need to understand the patterns of the microtubule cytoskeleton. In other words, now how does the cell harnesses or uses these properties of microtubules to define the distribution of microtubules in it? Okay, so that's what I'm about to illustrate in this wonderful animation. So everything starts with an MTOC, right? And from that, you have all the dynamics is at the plus end. You have growth of microtubules, boom. But then, if you imagine, they grow almost randomly in every direction. Now, some of them will hit the target, right, in the cell. And that interaction with the target tends to be a stabilizing interaction. In other words, it will inhibit catastrophes. So, by and large, so if you stabilize, but then the free ones, they still have their normal rates of catastrophe, they will undergo catastrophe. And at the end of the day, catastrophe tends to win. I'm sorry. So, but the good news is you end up with stabilizing the microtubules that are interacting, in a constructive way, if you will, with cellular targets. And this could be determined by a few examples. So, for example, here I'm just going to go through a couple illustrations of this idea. You have you transfect cells, insect cells, like S2 cells from Drosophila, with microtubule-associated proteins from, say, vertebrate neurons, like MAP2. You get this. So microtubules are forming a bundle. This is like a cross-section. You use another one like tau, which is a classic neuronal marker. You get a slightly different organization. And not going to go into too much details, but this could be interpreted as MAP2 binds in a certain way and then interacts with the microtubule with their plus ends, tau in a different way, and so on. What I want to emphasize is how, so the dynamics of microtubule is regulated by binding to proteins in cells, right? So this is in a way, it's similar to actin, but it's even more important in the case of microtubules because the intrinsic dynamics, the kinetics is almost irrelevant, right? There's just an excess of tubulin in cells, right? So everything has to do with this dynamic instability and the stabilization by interacting with proteins. So I like this conceptual example better. So I showed you the example of a microtubule being stabilized. But the beauty of the system is if the protein dissociates, now the microtubule will undergo a catastrophe, will be destabilized, and the regulation of this association itself can be regulated by kinases. This is a classic regulator. And here I highlighted MAP kinase, which is a signaling molecule. You're going to hear from, I forgot if it's with Carlos or Claude, but it's a very classic kinase involved in MAP kinase signaling, is the name. Or CDK. CDKs are classic kinases also implicated in the cell cycle. Now, the reason why I highlight this one is because you could see how if you phosphorylate in a CDK cell cycle dependent manner a protein which stabilizes the microtubule, it may lead to to catastrophe, to the removal of this microtubule. And that's going to be essential for the transition I mentioned, which is when cells have to enter mitosis, when they have to remodel the entire microtubule cytoskeleton to build a mitotic spindle, as opposed to maintaining the cytoskeleton, the microtubule cytoskeleton for migration, for example. Okay, so a very important regulator is these are proteins that bind the plus tips, right? Because this is where the business ends. So anything that can modulate that can stabilize, like I showed you, or promote polymerization versus catastrophes will strongly impact the microtubule cytoskeleton. So this protein here, EB1, is a classic plus tip binder. So here you see really nice images of cells where tubulin is labeled. I forgot if it's a label or if it's a fusion with a fluorescent protein here, but it doesn't matter, you would get the same. So tubulin in red, EB1 in green, and you see these dots of EB1, and they tend to reside clear here at the tip. So, and that's the tip that points outward, so that's the plus end. Okay? And in a movie form, now you see EB1 GFP for sure. No microtubule, but this is how it looks. Right, so this is very nice, this comet shape. So it's really showing that you have, let's look at this again, right? So what do you think this movement is due to? It's the growth, right? Polymerization. So somehow EB1 remains at the plus end in growing microtubules. Now, what did I just tell you about microtubule dynamics? They depolymerize. There are catastrophes. Do you see any catastrophe here? No. So we have a problem, because we have a plus end binding protein that is there, but only shows us polymerization. So what do you think could be happening? It helps stabilize, yes. Yeah, what else could be happening?
Is it like a target protein?
Like a what?
A target version for the stabilization tool?
I mean, in some ways, yeah, except it moves with the microtubule. So it's not anchored anywhere. So it does help stabilize the plus end. That is true. But I'm telling you, there are still catastrophes in this system.
It debinds when there's a catastrophe.
Exactly. Right? So if now you look at a so-called chemograph. So a chemograph is when you plot in one of the axes of the image time, right? So for example, here this is distance. So this would be, imagine you're looking at one microtubule, and you look at this microtubule over time. Okay, so the y-axis going down is the time, right? So you see this microtubule here. So when you see this, distance is going up over time, so it's growth. And indeed, when this microtubule is elongating, EB1 is bound to it. Now, EB1 is gone somehow. It's kind of not moving and growing again. And here you have the catastrophe. But you see, during this period, indeed, there's no EB1 signal. So that's exactly it. EB1 binds only on growing microtubule. And you could imagine ways in which this happens, right? Because the binding is dynamic, so it goes on and off. And if a catastrophe starts in an off period, then the catastrophe is launched, but it so happens that the EB1 can't bind a microtubule that's undergoing catastrophe. Okay, so so in this regard, this cartoon is a little bit well, it's not misleading. What this is showing here is actual polymerization. Okay, and so in the same way, you have proteins that bind the plus ends and promote growth. You have some that will promote catastrophe. Okay, so here is one of them, kinesin 13, and I'm going to get back to kinesin 13 because kinesins are molecular motors that can bind to microtubules and will eventually harness the energy of ATP to exert force on microtubules. And there's an emerging theme where these kinds of motors actually degrade the microtubules they walk on. In the case of kinesin 13, it's been known for a while because they bind to the negative end and their force basically pulls on protofilaments and triggers catastrophes. Okay, so that's what's depicted here. They induce a curvature, causing a catastrophe. They remain bound to the dimers, then detach, and the dimers can be recycled for the rescue. Okay, you have other ways to promote these catastrophes. So, because I told you the structure, what happens in a catastrophe is the protofilaments becomes curved, right? And that curvature antagonizes the stability of the whole microtubule. So they are bound not to sausages, but to statmins. I know I just said that to kind of wake you up. To statmins, and the interesting point here is, unlike kinesins, they are not motors. They do not exert force really to bend the filaments. They just have an affinity for the protofilaments, but the protein themselves are curved, right? And that's how they will trigger the catastrophe. And then, of course, as usual, the dimers will need to be recycled and contribute to the rescue. And statmin itself, interestingly, can be regulated by kinases, right? So you have many layers. And so that's similar to the previous slide I showed you, where you have signaling that can impinge on the microtubule dynamics by phosphorylating or changing proteins which otherwise bind to microtubules and so on. Okay, so I'm done talking about microtubule dynamics. Now I'm going to just give you a few examples of higher order understanding, and we're going to talk about motors. So, transport. I told you microtubules can be long, they can be stabilized by these interactions, and so they serve as tracks, long-distance tracks for long-distance transports. Yeah.
I wanted to ask what the difference between the centrosome and the MPFC is. Or the other difference.
the centrosome is an MTOC. So there are some MTOCs which, so again, microtubule organizing centers, which are not centrosomal in the sense that they don't have the centrioles. So there are cases where you have the gamma-Turk complex that can be positioned and trigger the formation of microtubules, but it's not a centrosome so proper. So the centrosomes are the most abundant in animal cells. Okay, so all the experiments especially focused on transport along axons. Now, if you think about a neuron, a nerve cell, you need quite a bit of transport because the nucleus is in the cell body. A lot of the synthesis still happens in the cell body, and yet neurotransmitters and all kinds of activity has to happen at the synaptic button, sometimes meters away, as we saw. Right? So you need transport, you need long-distance transport, and that involves microtubules, that involves motors that can walk and transport things along microtubules. So old school experiments involved injecting, I don't think anybody does this anymore, radio-labeled substances like amino acids in a cell body, and then eventually collect fractions along the nerve and basically study how those were being transported throughout. Now I'm just going to show you, so live microscopy has kicked in since, and this is still an old one. But what you see in this image, we're not going to linger here too long, but you see stuff moving, right? And by and large, it's moving up and down in this direction, and everything moves, from small vesicles, small particles, to even mitochondria. So these elongated things are not the Statmin sausages, don't get confused. They would be, yeah, this one is nice because the resolution is such that you see microtubules and you see these particles moving along the cables, along the tracks, okay? And then look at this one. There's one I wanted you to. This one is harder to see. Okay, so here you have some mitochondria warming their way. Some others are moving faster. So this one, look here. This guy is going, these guys are going. Okay, so did you see this? These two, they go in opposite directions on the same track. Okay, so we're gonna have to solve that because so there are a few, you know, it's a simple observation, but there are a few questions. How things, well, first they move on tracks. So you're gonna have to think about associating a motor, something that can walk on the track. I'm giving it away, that's gonna be kinesins, dyneins, and those are going to have to be able to carry a cargo, be it a vesicle, be it an organelle, and so on. Okay, so the point I tried to highlight was that you have, on individual tracks, you can have particles moving one way, particles moving another way. Okay, so you don't really see that on actin filaments. Well, first, they are thinner. It's harder to image than for the microtubules, but you would not really see movements in both ways on actin filaments because mostly it's myosin, and myosin tends to work mostly in one direction. Okay, so here this is suggesting that you have motors that can work in either direction, even on the same filament, on the same microtubule, presumably because they are big enough. So in still form, so you have organelles. So this one could be, I mean, I don't know what this particular one is, but you can, in other words, you can have relatively big cargoes, right? Moving all the way to the tip of the axon on these microtubule tracks. And that's called anterograde movement. So it's very analogous to the anterograde versus retrograde we talked about in the secretory pathway. It's pretty much the exact same thing, and it also involves transport of vesicles. Sometimes it moves entire organelles, And now you know it involves tubulin, and it involves the motors I'm about to tell you, kinesins and dyneins. Kinesins are responsible for the anterograde transport because they work towards the plus end. Dyneins, retrograde transport because they work towards the negative end. Okay, so those are some of the key principles. So indeed, when you look at individual particles, so this is going to be shown here. Boom. So individual particles, they do not hesitate. It's not like undergraduate students in the corridors. They don't go and then, no, I have to go this way. No, they know where they go. Okay? Or professors sometimes. And that's because they are bound to a single motor. Okay, so basically the directionality is given by the motor, and once a given particle, a given vesicle is associated to a motor, it goes in one way or the other. So the first motor we're going to briefly talk about is this one, kinesins. So I'm showing you a lot of details, but mostly I'd like you to get the essence of it, which is you have these globular heads, which is an important part. They are the ones to bind ATP, the nucleotide, so they can hydrolyze ATP. That means they can harness the energy of ATP, undergo conformational changes, and they're also the ones to bind to microtubule. Okay, just like, similar to myosin. So that's the part that can walk on microtubules. To have the walking, they have to dimerize. So that involves this linker and stalk, and the stalk eventually will end up with this tail, and that's going to bind to a cargo, right? So this is the essential basic organization I need you to sort of get from this. And it's almost identical to myosin in this regard, okay? And this cross-linking, so there are really cool movies on the internet. I encourage you to go check them out, showing models of literally kinesins walking on microtubules, carrying cargoes. And that means on the cargo, if the cargo is a vesicle, you need to have a receptor to interact with the kinesin molecule. So you have some specificity. Yes?
Isn't the vesicle like 50 times its size, though? Or like much larger?
Yeah, it could be much bigger.
So how does it stay down so securely?
That's a good question. I'm not sure, actually. I mean, I could say, well, the affinity for it is really high. It's regulated by phosphorylation. That's a good question. So the thing is, okay. No, it's a good question. Now, I'm not sure it's only one molecule per. So I would actually say, and I need to look this up, but I would invoke multiple molecules of kinesins per vesicle, per organelle. I'm not sure one molecule can transport the whole organelle, especially you have a viscous drag in the cytosol, etc., etc. And then some of them move fast, as you see. Yeah, that's a good point. But anyway, they walk towards the plus end, and this is all due to this inherent structure of the globular heads and their ability to bind to, here in this case, the beta-tubulin subunit. So this is also analogous to what I said about actin, which is, at the end of the day, you have very specific protein-protein interactions that are labile, but that are nonetheless specific, and so you have to have the structures match. So the walking is extremely stereotyped. Okay, it's not like us on a curb in the street. You have to have perfect match. So then you have this kind of cycle connecting the hydrolysis of ATP to the actual movement. So you start here, let's say, ADP bound and not bound to the microtubule. Upon releasing ADP, there's a conformational change that allows binding to the microtubule. Now this forward head moves, binds ATP, there's a big change of conformational for the forward movement, and so on and so forth. Sorry. So you can look this. You can look. There's. Sorry, I sound like I'm bored because I am a little bit. Because I just can't fake it. It's just the basic concepts I need you to get out of this. Is not the details of what happens at what step. Is that is very similar to meiosis. Is that you have by and large in the entire cycle, you have a coupling between the hydrolysis of ATP and the forward movement, which is exerting mechanical force. So in thermodynamic terms, that's the coupling. You harness the energy of an hexergonic reaction hydrolyzing ATP with an endergonic reaction, which is exerting force, making, having a movement. And that is the way this is achieved is through coupled conformational changes throughout this cycle. But just like for myosin, you don't have a direct coupling between the energy of ATP and the movement. Yeah.
So each time there's a conformational change, does the coil more?
Not necessarily. I mean, quite frankly, I would be hard pressed to tell you exactly, where the energy goes. Let's say when you hydrolyze ATP, exactly where the energy goes at every step, it's not entirely clear to me. And I'm not sure it's entirely clear to anybody, right? Because that would mean you would need to be able to quantify precisely each conformational change and determine the free energy, the change in free energy that goes with each. Right? So my point is that you have a coupling at the level of the whole cycle, not necessarily at the level of each individual step. And I kept talking about the parallels with myosins because I think for you guys is a key aspect to sort of remember is that in all of these cases, in both of these cases, you have structures that have evolved to hydrolyze ATP and couple that hydrolysis through a relatively complex cycle of conformational changes and binding to cytoskeletal elements and binding to nucleotides, but to basically convert the energy of ATP into mechanical force. And somehow, structural analysis, so this is not easy to see here, but what this is illustrating is that the two domains, they end up resembling each other structurally with these beta strands here and these helices and so on. But they evolved independently. They are not related molecules, evolutionarily speaking. Right? So kinesins and myosins evolved independently and yet ended with a very similar solution to couple hydrolysis of ATP with movement on a cytoskeletal track. Then the other business end of kinesins, of course, is to bind to cargo, and this we would now assume this binding is extremely stable. That's a good point. To move cargoes along always towards the plus end. Some of them are hetero-trimeric, and they also have these light chains here. And then other kinds of kinesins. So this one is extremely important, kinesin 5. It's bipolar. That means it can bind to microtubules on either end and help them slide against each other. So can you think of a place where you would have microtubules being anti-parallel and in need of positioning each other? Yeah. Actually, in the muscle, this is actin.
Oh, yeah.
Yeah, it's actomyosin. Here, microtubule. Yeah, in the mitotic spindle, exactly. So kinesin 5 is important for this to help organize and position the spindle. And kinesin 13, we saw it, it's mostly the heads, because there's no cargo, it's more about bending protofilaments and causing catastrophes. All right, so next one is our dynein motors. I mostly have cartoons about those, and the logics are somehow similar. But in structures, it's more separate. In other words, you have an ATPase domain. This happens to be the head. And it's connected but separate from another domain here called the stock, and that's what binds the microtubules. And finally, the stem here is what will bind a dianactin, which is an adapter for cargo. But by and large, you have the same basic principle. An ATPase domain, a domain to bind the microtubule on the track, and then something to bind to the cargo or wherever you need to. So here too, hydrolysis of ATP is coupled with this conformational change, which can move things around, right, like this. Get that? Okay, but again, same logic, coupling ATP hydrolysis done in one domain, bound to microtubule and converting the energy of ATP into force. And here the adapter is this dianactin, which is kind of complex, but this is the adapter to bind to the cargo, right? So I don't need you to worry too much about this. This is more the part I need you to worry about. Not worry, don't worry. Focus on, because that's really important for the organization of the cell, right? Because again, for actin, you can get away with myosins that can. Actually, let me make you think about this. Why can you get away with only one kind of motor, myosin that walks in one direction when you have actin? Yeah.
You don't necessarily need to form at the center of the cell, from the center of the cell.
That's right. Because that's exactly right, actually. Because actin can be plus, minus, it doesn't. The orientation is much more variable. Here, by and large, you have this organization, right, where the plus ends are always towards the outside, the negative, and towards the inside. So if you want to move things in either direction, and we saw in the secretory pathway lectures that both anterograde and retrograde transport are essential. And by and large, this coincides with inside of the cell versus outside of the cell. So which motors would you use for anterograde movements mostly? Kinesins and dynesins for retrograde movements, right? So now you see why I'm deeming this slide more important, because it helps integrate the different lectures and hopefully makes sense for you guys. Why that if you organize your microtubule in this way, you really need two kinds of motors in order to achieve both types of movements, anterograde or retrograde. Okay, I'm gonna spend the last few minutes. What's this? Sorry, I thought it came from the. Okay, so intermediate filaments. I mean, you know, it's also Monday morning after the spring break for me too. Okay, so intermediate filaments, they are very important. Like keratin, you know, where you have, for some people, it's really important. This is keratin. This is intermediate filaments. Mine is getting gray. Doesn't have anything to do with intermediate filaments. But, so they are really important structural elements, and they are very diverse, okay? So here are a few examples of them in cells. And as you see, they kind of reach out all across, and they're extremely stable compared to microtubules, compared to actin. So they contribute to the physical integrity of cells over time, right? But they are somewhat less dynamic, right? And they are not so much less dynamic, not polarized, and they don't serve as track for known molecular motors. So like I said, it's more about cell and tissue integrity, like this, the scaffolding. And in part because they have this great tensile strength, that means you can pull on them quite a bit. So mostly mechanical mechanical resistance, so they are there for physical properties themselves. And indeed, part of this comes from the fact that even the monomers are fibrillar proteins. So here I'm showing you a cartoon. Well, so this is not a cartoon, it's a fuzzy electron microscopy image, but the cartoon representation would have globular domains at either N and C-terminal ends. So that would be for interactions with surrounding structures and other dimers. But then, more importantly, this rod domain here, which is providing the tensile resistance, okay? And now that's the well, that's more like a dimer at this point. But then those can further polymerize and create higher order fibers. So that's what's showed here, where you have now, again, electron microscopy. So you see some structure, some regularity, but you now have a tetramer that forms this filament and these protofibrils. And now you have great tensile resistance here. It's like a rope, okay? So that's the basic organization, the basic structure, and so that's going to be the final slide of today's lecture. Like I said, you have a diversity of those. They're important in epithelial cells. Keratin ends up accumulating in cells in our skin, which dies and ends up forming most of the dust that is everywhere in New York City. I mean, it's not just human keratin, it's also rat keratin, quite a bit. And then those are really important desmins, for instance, or vimentins, because they are at desmosomes, so integrity of epithelia, not just based on keratin which goes across in the cell, and that links these desmosomes. So integrity of cells, but integrity of tissue. Entire desmos. So you have this mechanical coupling between cells within a tissue that happens through these desmosomes. And then finally, the last two, neurofilaments, again, just specialized for neurons, again, because axons can be very long. You need physical integrity there too. And the final one is actually extremely important, maybe under-covered in this. I don't know, did you guys hear about lamins in molecular cell 1?
Yes.
Yeah, because they are very important for gene regulation just underneath the nuclear envelope. You did. You're not faking that, right?
We did with Paul.
You.
Paul.
Oh, yeah. So, of course, you must have heard about all the glory of lamins. Anyway, so indeed, lamin. Cheap sick of lamin?
No, that's high C.
Okay. Okay, so you heard a lot about lamins, important for nuclear integrity, important for mechanical coupling, and for the cell to respond through gene expression to mechanical forces. And I'm going to stop here.